very much for this introduction. It's great to be here to speak at my first grand rounds here at Yale. So I think that for some of you, much of the work that I'm going to be presenting today, you may have already heard during the talks that I gave during my interview process. So I apologize for those of you who have already heard some of this work. I'm going to tell you about the studies that I did during my postdoctoral fellowship to better understand the mechanisms of lung tumorigenesis by mutant EGF receptor. And so just as a starting point, we all know that lung cancer is the most common cancer in the world. And in 2010, close to 160,000 people will die of lung cancer. So it's really the leading cause of cancer death in the United States. The 5-year survival for all stages of lung cancer is about 15%. And these numbers really tell us that we need to develop new strategies for the early detection and the treatment of lung cancer. And lung cancer is not just one disease. It actually can be subdivided into two histopathological categories: non-small cell lung cancer, which makes up about 80% of all lung cancers, and small cell lung cancer, which makes up about 20% of these. Non-small cell lung cancer itself can further be subdivided into large cell carcinomas, adenocarcinomas, and squamous cell carcinomas. And today I'm going to tell you about a lot of the work that has been done to study the genetics of lung adenocarcinomas. And this has really taken off during the past 5 or so years, in which large sequencing studies have been done to see which genes are mutated in lung adenocarcinomas. And here is a slide to summarize the mutational analysis of 623 genes that was done in close to 200 lung adenocarcinomas as part of the tumor sequencing project carried out by the National Cancer Institute. And some of the findings that it's important to highlight from these studies are the genes that were known to be mutated in lung adenocarcinomas, like P53, KRAS, EGFR, were found to be mutated at the expected frequencies. However, genes that were not really previously known to be mutated in lung cancer were also found at quite reasonable frequencies. And I think that in total there were about 26 of these 623 genes that were found to be mutated in multiple samples, really indicating that they probably are involved in the tumorigenic process. One of the other important findings that emerged from these studies is that there are actually different classes of lung adenocarcinomas based on these mutations. So you don't ever see tumors, lung adenocarcinomas, that have mutations in KRAS and mutations in EGFR, for example. These are mutually exclusive, or used very, very, very rarely do you see them together. On the other hand, mutations in the tumor suppressor gene P53, which are found in about almost close to 50% of lung adenocarcinomas, are found in tumors that have KRAS mutations and tumors that have EGFR mutations. And so one of the questions that emerges from this is really, what does it mean that a tumor only has a KRAS mutation or an EGFR receptor mutation? Does this make, is there a clinical relevance to this? And taken together, all of these studies have really highlighted the importance of the EGFR, ERBB signaling pathway for lung adenocarcinoma formation. And in this diagram, what I'm doing is I'm just highlighting the EGF receptor pathway. So here is the EGF receptor or other family members of the ERBB pathway, and downstream signaling pathways, the RAS pathway and the PI3 kinase pathway. And right here, the genes, highlighted the names of the genes in these signaling pathways, and in red, the frequency with which this gene is mutated in lung adenocarcinomas. And you'll see that most of these genes are mutated in this disease. So what I'd like to tell you about is the work that I focused on, which is really to study EGF receptor mutant lung cancer. And these are images taken from the lungs of a lung cancer patient with a tumor, and then treated with an EGF receptor tyrosine kinase inhibitor, ERESA, where you see this really rapid response. And so at the beginning, it really wasn't understood why there were some patients who were coming to the clinic who were having these really dramatic responses to these drugs. And several groups amongst the groups of Tom Lynch when he was at MGH, other groups in Boston, and our group at Sloan Kettering, found mutations in exons encoding the tyrosine kinase domain of the EGF receptor. And in this diagram, you can see a schematic representation of the EGF receptor TK domain with depicted the two most common mutations that are found in lung adenocarcinomas. They are point mutation in exon 21 that leads to substitution of an arginine for leucine at position 858, and small in-frame deletions in exon 19 that lead to elimination of an LREA motif. And these mutations are associated with sensitivity to tyrosine kinase inhibitors like gefitinib and erlotinib. And so here is just to show you the clinical data, and show you how significant the responses are. If you look in EGF receptor mutation positive lung adenocarcinomas, lung tumors, you see about a 70% response rate upon treatment with gefitinib, in this case gefitinib, versus about a 47% response rate upon treatment with chemotherapy. If you look at tumors that are negative for EGF receptor mutations, you see that the response rate to treatment with tyrosine kinase inhibitors is very low. And so when these mutations were discovered, what I wanted to do was I really wanted to study the role of these EGF receptor mutants in lung tumor initiation and in lung tumor maintenance. And so I decided that the way to do this was to express these different oncogenes specifically in the lung epithelium using a tetracycline-inducible transgenic model. And the reason to use a TET-inducible system was that if we take mice that express that carry mutant EGF receptor under tetracycline-inducible promoter, cross them to mice that express RTTA, so the reverse tetracycline transactivator under the control of a lung-specific promoter, and by transgenic mice, if we feed them doxycycline, we can turn on expression of the gene. One of the nice things about this is that you can reverse the whole process because you can withdraw doxycycline and shut off expression of the transgene. And what we see when we express these EGF receptor mutants is that the mice develop lung tumors, and we can use imaging, MR imaging, and then look at the tumors histologically. So we were able to show that these tumors, that these EGF receptor mutants could initiate tumorigenesis, and we were also able to then, by withdrawing doxycycline, so shutting off expression of the transgene, show that the tumor went away, suggesting that the tumor cells were dependent upon continued expression of the transgene for their survival. And similarly, when we take mice with tumors and we treat them with a tyrosine kinase inhibitor like erlotinib, the tumors shrink and go away, very much recapitulating the scenario that you see clinically in patients with EGF receptor mutant lung adenocarcinomas treated with tyrosine kinase inhibitors. So this is all great, but the problem is that patients who are treated, who initially respond to treatment with these drugs, almost inevitably develop acquired resistance to tyrosine kinase inhibitors. And these are images of the lungs from a patient who was treated with a tyrosine kinase inhibitor, initially responded, and then the tumor began to grow back. And so a number of groups, including our group when I was at Sloan Kettering, found a secondary mutation in EGF receptor in about 50% of drug-resistant tumors, and that accounts for resistance to tyrosine kinase inhibitors. And so what we went ahead and did when we were when we discovered this secondary mutation is we generated transgenic mice that express these mutants, the EGF receptor with the sensitivity mutation and with the drug-resistant mutation in cis in the same transgene, mimicking the human situation, this human scenario that you see with acquired resistance. And so what happens is if we take these mice, and this is a lung tumor here, and we treat them with erlotinib, we see that the tumor doesn't shrink. And this is in direct contrast to one of the mice that has an L858R-induced tumor that is about the same size as this tumor here, but within a week of treatment with a tyrosine kinase inhibitor, this tumor has really gone away. And so we were able to model T790M acquired resistance in vivo. And importantly, one of the things that we can do with these mice is we can actually use them to see if we can develop new strategies to overcome resistance due to the presence of the T790M mutation. And we tried initially, this is work done in collaboration with William Pao, and who was at Sloan Kettering and now is at Vanderbilt, and Lucia Regales from Williams' lab. And what we saw is if we take these mice and we treat them with an irreversible tyrosine kinase, BIBW, we do not see tumor shrinkage. However, and if we treat them with an antibody to EGF receptor, cetuximab, we also in this case do not see the tumors go away. But interestingly, the combination of both of these drugs is very powerful, and we see remarkable responses in these transgenic mice. So the combination of BIBW plus cetuximab, suggesting that this may be a way of overcoming T790M-mediated resistance. And the importance, this really has formed the basis for a clinical trial that is actually going to be starting at Memorial Sloan Kettering, at Vanderbilt, and at Yale in patients who acquire resistance to tyrosine kinase inhibitors. And so hopefully we will be able to follow this in the clinical setting. And so one of the things, some of the experiments that we're currently doing are actually to take these mice that have responded to cetuximab plus BIBW, and we are now treating them for doing long-term treatments in these mice to see whether we see drug resistance emerge with a combination of cetuximab plus BIBW. And the hope is that, well, the best thing would be that drug resistance does not emerge. But if drug-resistant tumors do emerge, what we hope to be able to do is we hope to be able to analyze the tumors that resistant tumors that emerge in these mice and find out why the tumors are resistant, and then predict what may happen in human patients who are being treated with this combination of drugs. So that is sort of the vision for some of these studies going ahead. And I'd just like to point out in this table, which is very complicated, but if you look at the circles, these are all different mouse models of lung adenocarcinoma that have been induced by different oncogenes, and these are all sort of, this is a summary of all the different treatments that have been done, and this is a collection of work of several groups, including especially, well, our work, but also Kwok and Wong's work at the Dana-Farber. And what you can see is that actually using these mouse models, there have been several strategies to overcome resistance to tyrosine kinase inhibitors that have been identified using these genetically engineered mice and that are being sort of looked at to design trials in humans. I'd just like to very briefly show you how we think that we actually can model acquired resistance to erlotinib in the mice. Also, because not only by generating transgenic mice that express genes that are already known to be mutated in cases of acquired resistance, but we can actually start off by, we can also get spontaneous resistant tumors. And here we devised a protocol in which we take mice that have lung tumors, we treat them with drug, and we found that we have to treat them in multiple rounds of treatment. And so we see initial responses, treat them for various rounds. Usually by the end of the third round of treatment, we see drug-resistant tumors emerging. These are L858R or exon 19 deletion expressing mice treated with erlotinib long term. And we have seen in tumors that we've taken from these mice, we have seen the T790M mutation, we have seen MET amplification, all mechanisms of resistance that have been seen in the human situation. And so now we're analyzing the tumors in which we did not find T790M, in which we did not find MET amplification, and we're analyzing these to see if we can find new mechanisms of resistance to tyrosine kinase inhibitors to try to make some headway in this, let me just go back, in this pie chart to find out what these 40% of TKI-resistant tumors, what the mechanism of resistance is in these cases. And one of the strategies that we are using is we are performing exon capture and high-throughput sequencing of about 2,000 mouse genes. And so we take the genomic DNA from the drug-resistant tumor, we hybridize them to chips on which we've arrayed exonic sequences for these 2,000 cancer-related mouse genes, and then we elute these, and we are performing high-throughput sequencing. We actually have already sequenced 10 drug-resistant and 4 untreated L858R-induced tumors, and we have been in the process of trying to call single nucleotide variants for about 6 months now. It's been a very laborious process, and I think every time we're getting there, we're getting very close, but it's been quite a struggle. There's a lot of informatics work that has been quite a struggle for these studies. Importantly, though, we have been able to, in some controlled tumors that were analyzed, we have been able to detect the T790M mutation. We also had one tumor that had a KRAS, spontaneous KRAS mutation, and we were able to find that. So it looks like the system is working, but we have, unfortunately, we're calling more single nucleotide variants that actually validate. So when we go to validate, we only have about a 10% validation rate. So our bioinformatics pipeline still needs some work. So what I've told you about today is I've told you about some of the studies that we're doing to study, to use these mouse models to understand different molecular subsets of lung cancer and to understand mechanisms of drug resistance. There are other areas in which we hope to be able to make headway using these genetically engineered mice, which are in the study of metastatic disease and work early detection on the early detection of lung cancer. And of course, we'd also like to understand which genes cooperate with some of these different oncogenes that are known to be driver mutations in lung cancer. And in particular, one of the ways in which we're addressing this question is to better understand the biology by which mutant EGF receptor transforms the lung epithelium. And we know that EGF receptor is part of the ERBB family of receptors, and we've designed some studies to try to understand the role of heterodimerization by mutant EGF receptor with either ERBB3 or ERBB2 in tumorigenesis. And so what we have done is we've actually crossed our mice that express mutant EGF receptor and RTTA to mice that carry a floxed allele for ERBB3 and that also carry a transgene for TETO-CRE. And so we started out by looking at what deletion of ERBB3 would do, because there was quite a bit of evidence in lung adenocarcinoma cell lines with human lung adenocarcinoma cell lines with EGF receptor mutations that deletion that ERBB3 signaling through the PI3 kinase pathway was important for signaling to the PI3 kinase pathway went through ERBB3 in these cell lines. And so we deleted, so using this system, what we can do is we can delete ERBB3. And so we take mice that have all of these transgenes in the absence of doxycycline, have wild-type ERBB3, and do not express mutant EGF receptor. The moment you add doxycycline, you turn on expression of mutant EGF receptor, you delete ERBB3 in the same cells in which you've expressed mutant EGF receptor. And in these Western blots, these are diagrams just to show that actually we do delete ERBB3. So this is by genotype when you should have ERBB3 and when you shouldn't have ERBB3 in the lung, you don't see it. So the system is actually working, but you do have expression of mutant EGF receptor in both scenarios. And interestingly, these are data from the L858R mice. In all of the cohorts of mice, we see that deletion of ERBB3 does not have an effect on tumorigenesis by mutant EGF receptor. When we look histologically, the tumors that develop are similar in the presence and absence of ERBB3, and also signaling pathway activation. So here's phospho-ERK, here's phospho-AKT, does not change whether we have ERBB3 or whether we don't have ERBB3, whilst we do always have similar levels of mutant EGF receptor in the lungs of these mice. Importantly, we were able to do a control in cell lines just to make sure that in the transgenics we express human mutant EGF receptor. And in the mouse, though, we're deleting mouse ERBB3. And so what we wanted to do is we wanted to make sure that actually we can get dimerization of a human EGFR and mouse ERBB3. And when we transiently transfect cell lines, if you look at this lane here, you see that when you immunoprecipitate with an antibody to ERBB3, so you pull down ERBB3 and you also pull down human L858R. So we can. So the results that we're seeing in vivo are not just because of an artifact. When we use the exon 19 deletion mice, we also see that tumors develop in the absence of ERBB3, and we get very good deletion of ERBB3 here. Similarly, signaling pathway activation is similar in the tumors and really depends more on the levels of EGFR than on the levels of ERBB3. Importantly, one of the things that we have found, and these are MR images of mice from either L858R mice or exon 19 deletion mice in which we've deleted ERBB3, what we see is we see that the tumors do go away, and this is a really massive tumor here, so a mouse treated for a month with erlotinib, the tumor goes away even though it arose in the absence of ERBB3, indicating that really probably mutant EGF receptor is signaling, doesn't require ERBB3 for tumorigenesis and nor for the response to treatment with tyrosine kinase inhibitors. So to summarize, these results, the lack of ERBB3 does not prevent lung tumor formation in EGFR, L858R, and EGFR deletion mice. And now we're trying to address a lot of the biochemical aspects of this work now that we've completed the in vivo work. So we're trying to really understand how active the PI3 kinase pathway is in these tumors. How does mutant EGF receptor dimerize? Does it homodimerize? Is it dimerizing with ERBB2? And in these tumors. So in conclusion, I'd just like to acknowledge the people I worked with through all of these studies. My mentor, Harold Varmus, who's now at the NCI, people who are still transitioning from Sloan Kettering to the NCI, and we're in the Varmus lab, Pang, Udayan, and Will Lockwood, who have helped on various aspects of these studies. The lung cancer oncogenome group has been a great group to work with at Sloan Kettering, which is a group of thoracic oncologists, surgeons, and scientists who we've all worked together to sort of tackle some of these problems. William Pao, now at Vanderbilt, and collaborators at Cold Spring Harbor and MIT, with whom we're working on this high-throughput sequencing of drug-resistant tumors. Thank you very much, and I'll be happy to take any questions. This is the Varmus lab. There you go.